I thought I'd say just a few words, because I haven't met most of you before, about who I am and how I got here, and then talk about my first impressions of the Dana-Farber, having been here for now a little bit more than 3 weeks. But I am really coming home because I grew up in Brookline and in Newton and have spent virtually my entire career at Harvard, at one institution or another. I went to Harvard undergraduate and Harvard Medical School, and then did my internship in medicine at the Massachusetts General Hospital and my subspecialty training in rheumatology at MGH. Now, rheumatology is just the opposite of cancer. The immune system is overactive as compared to being underactive in cancer. Two halves of the same coin. My dad was chair of orthopedics at Mass General and then chair of orthopedics at Children's Hospital, and I worked in his lab, so I got to know both those places as well. And then I came back from a postdoctoral fellowship training in immunology at the NIH, and I came back to Harvard and sort of rose through the ranks as an immunologist and a physician. I attended at Brigham and Women's Hospital, so that adds another hospital to my list, and had my laboratories at the Harvard School of Public Health, so I feel like I really covered virtually all of the Harvard institutions. And now we're 3 generations at Harvard Medical School and Massachusetts General Hospital, since my older son is training to be a cardiothoracic surgeon at Mass General with an emphasis on thoracic oncology. So all this was wonderful, and I'm a Bostonian, definitely a Bostonian, but I did take a hiatus for 4.5 years and went to New York City to be the dean at Weill Cornell Medicine, which was a fabulous experience, and it's a fabulous place. But when I got a call from the chair of the board, Josh Bekenstein, about a year ago, asking me if I were interested in coming back and considering the presidency of the Dana-Farber, I just couldn't resist. Because this is such an amazing institution. And of course, I knew the Dana-Farber when I was here at Harvard, and in fact, I started my research career at the Dana-Farber. When I was a 4th-year medical student, I spent the year in Harvey Cantor's lab, learning immunology. So to me, this is like a big circle. It's where I started and where I'm finally coming back. And I'm really honored to be the CEO and president. I follow a wonderful man, Ed Benz, and he has big shoes to fill, and he's did a terrific job. So, we're all, I know, very grateful to him. So I've been here for about 3 weeks. Actually, I shouldn't say just 3 weeks, because I actually did the PMC, rode 86 miles, never having done much cycling before. I was determined to get through that 86 miles. And then joined a large group of volunteers doing the Jimmy Fund Marathon Walk. So I feel like I had a very good introduction, an athletic one, but a very good introduction to the Dana-Farber. And I'll tell you, I was so inspired by the passion, the enthusiasm, and the support as we rode our bikes or did the walk. The hundreds of people that turned out in support of us, and the hundreds of people that walked or biked or were volunteers. I think this is an incredible community, and that Dana-Farber arouses incredible support because of what it does. One of my, I think, most profound impressions when I came was, even though I knew how excellent the faculty were at the Dana-Farber, but actually spending the first month I was here in September and the last 3 weeks meeting with so many of the faculty, the chairs of departments and chairs of division, to realize the quality of the faculty. We are a fairly small cancer institute compared to institutes like MD Anderson or Memorial Sloan Kettering, But I would say that the quality of the research and the quality of the clinical care that we deliver is unparalleled anywhere. And I know those institutions well. So it's just a pleasure to have been here this morning and heard the absolutely stunning presentations given by 4 individuals from our faculty and the overview by Barrett Rollins. And this is truly an amazing time for cancer research. It is an amazing time. Last night, somebody asked me a question about preventive medicine because we were talking about precision medicine and how that really started here and how we are on the cutting edge for that. And we talked about cancer immunotherapy, which is my own particular interest, and how that is eventually going to be precision immunotherapy as well. But what we didn't talk so much about was precision prevention. And I think, our ultimate goal is to prevent cancer in the first place. That's the best solution that we have. And we need a lot of research to figure out how to do that, because we haven't figured out how to do that yet. There's one cancer vaccine that's been approved, Provenge, for prostate cancer, and now that company has folded because the vaccine was not very successful. But the precision prevention view is, the right strategy for the right person, the right family at a time. And the science of prevention is not just, eat your vegetables, stop smoking. It really is genetic risk assessment for families, for patients and their families. And, we've recently started a community effort for the one vaccine we have that can prevent cancer, and that's human papillomavirus. Nobody anywhere no women should be developing cervical cancer. We have a potent vaccine, and it is not being used. Only 40% of American girls and boys are receiving this vaccine, and it's less good in other countries. Better in some, but less good in others. So we're taking steps here, and I just wanted to highlight a couple of those because I haven't spoken about them before. We're extending the Profile program, which sequences all of our patients' tumors, a pretty amazing program, to the germline, to actually look at the DNA in your normal cells, all the DNA in your normal cells, as well as the DNA in the tumor. And we're investigating germline mutations in acute lymphocytic leukemia cells. We need to figure out who is at risk for getting cancer in the first place. And this came up in the thoracic oncology visiting committee session that I was in. Is some of this genetic? Is some of this inherited? And there are some cancers that have a familial tendency. And we need to know that ahead of time. So we need to look at the families of patients with cancers that can be hereditary and make sure that we prevent that. And I heard a moving story from you just before lunch, Susan, about how that strategy has worked for you. So I did want to say that. So I've had a tremendous number of meetings with people. They've been really great. I'm just impressed, as I said, and I want to say again, by the quality of our researchers and the quality of our clinicians, really unparalleled. And to me, everything that we do at Dana-Farber is focused on our patients. And whether we do it in the laboratory by trying to find new treatments for cancer, or whether we do it at the bedside, that is the heart of everything we do, and that is sacrosanct to me, preserving the unbelievable quality of care we give our patients. And I've heard that from so many of you, and from others. Everywhere I go, Dana-Farber is just the place to go to get the most compassionate, highest quality patient care. We have challenges, and we have opportunities, and sometimes the challenges turn into opportunities. So we are in a rather tough healthcare environment with downward pressure on healthcare cost reimbursements, as you all know. And given At the same time, the compression of funding from the government, from the National Institutes of Health, whose budget for science has decreased by 25% over the last decade, we do need to search for other means that will allow us to continue the research you've heard about today. One way to do that, of course, is to partner with the private sector. And I think, as I said last night, I think this is critical to do because it's critical for our patients. What's the quickest way that we can get a discovery from the laboratory into a new therapeutic for our patients? Well, academia is terrific. We are terrific at basic science discoveries, and we're terrific at translating those discoveries in preclinical animal models and even in human cells, as you heard also today. When it comes to doing phase 2 and phase 3 trials, we need the help of pharmaceutical companies because they do those very, very well, and they're extremely expensive. And so partnering, getting pharmaceutical companies to support our research by sponsored research alliances, which we have and which we should increase, is a good thing. And as I said, it really accelerates the path from bench to bedside. Our faculty is terrific at getting grants from the NIH, and actually, we had an 8.7% increase in grant funding from the NIH, even in a time where NIH funding is static. But that's not going to cover the brilliant research and the risk-taking research that you heard about here. It's always been my philosophy that if you're going to be a scientist and you are passionate about what you do, you have to take risks. Because 90% of experiments fail, grants get rejected, papers get rejected, but if you have the fire in the belly to want to cure a disease, to find better treatments for a disease, then you have to think big. And when I talk about what my impression of Dana-Farber is, it is we have faculty here who think big, and they can do that because they have the kind of support that you can't get from the NIH, that you can't get from many private foundations, but that you can get from generous donors who understand how important it is to take risks. So I really want to thank all of you very much for your support and your commitment to this institution. I am thrilled to be here. I think the future is very, very bright for cancer, very bright for cancer. It's not going to be as easy as curing HIV/AIDS, because that's just one virus. Cancer is 1,000 different mutations. And we have to figure out how to overcome the resistance of that tumor cell to the first therapy. So we have some therapies that do amazing things, and they last for months or maybe even years, but a tumor's pretty smart, and it can evade that. So I think the next frontier for precision medicine is figuring out how to overcome and circumvent that resistance. The next frontier is figuring out how to always activate the immune system in all tumors. And that's going to take some daring research. Ultimately, the solution, I think, is the solution that worked for HIV/AIDS, which is combination therapy. And I said that last night, and I'll say it again, because I think it's so important. But to do those kinds of experiments where you are rapidly testing combination of drugs in human ex vivo models, and those ex vivo models are being developed right here at Dana-Farber Cancer Institute, that takes a lot of time, it's risky, but ultimately I think it's the way to go. So we've got a lot to do. I don't have a detailed strategic plan yet. I'm going to do a lot more listening and talking to all of you, to the trustees, to our faculty and staff about what they think the major challenges are and the major opportunities are for Dana-Farber. But it's wonderful to know when you're heading an organization that the people you're leading are A+++. So thank you so much.